this one growing horizontally off that colony there?
It does look that way. Take a look.
All right, Marley, go ahead.
So you have these white ones. You have the white kind of plate-looking one in the back with the holes, all the pores in it. You have one of the heteroconies in the background. And then in the foreground near the bottom, you have a clear thing, which is a tunicate. It's got some debris on it.
Mm-hmm.
But you have at least 3, probably 4 species of sponges here and a tunicate.
Would this be a good example for a 3D model of different species?
I think it's pretty small, though.
Yeah, it's pretty small. But...
Okay. It seems big compared to everything else.
They did do a heteroconia, a large one, earlier.
Okay, then never mind.
Is that a no to the...
Yeah, we won't do a 3.
All right, go ahead and come away.
I think so, yes.
Roger that.
Oh, you're right. They did that at 10:30 UTC. So sort of towards the end of the last dive.
That's why I saw it, because I wasn't awake at the beginning of the last time.
Yeah. Good on you for reading the notes.
I actually watched it. What could that fish be?
Ben?
Long nose.
Male or female?
No claspers, so female. Have
Right.
you seen any males?
Wow.
I have not seen any today.
Ben, you're going to be a long-nosed skate expert by the end of this.
They really need a monospecific taxonomic expert, just one species.
Just think how full the ship would be if you had that for every critter out here.
We need a bigger boat.
Yeah, bigger.
Just lately. A cruise ship. Coming right at us.
She's coming straight to, coming right at us.
First one I saw inside its mouth. Wasn't yellow though.
Oh. No, that's a black gill rockfish.
I'm gonna reset you really.
Thanks, Ben.
Yeah, we're gonna be slightly downslope, it looks like, for about 100 meters.
Roger that.
Probably 200 meters downslope.
Yeah, the slope map shows that it like gets pretty flat here.
Right. Roger
Shall
that.
see. Problem. Are you fighting the current here in this direction a bit?
Yeah, a little bit of a bit of a current, but not too crazy.
Well, you're doing a great job, even when you're going down slope.
Thank you. Hopefully we can find you that blue cheese. I was gonna say blue cheese baby because it goes blue jeans baby.
Well, if
blue
you find
cheese
it, you
baby.
definitely can call it that. Revise the common name. How large do those things grow? The blue cheese.
Probably less than 10 centimeters.
Roger.
Yeah, can we get the lasers back on?
Yeah, sure.
Thank you.
Mhm.
Definitely not many corals out here.
So what makes this a favorable sponge habitat but not coral habitat? Presumably they both like flow.
Yeah, it could be the sedimentation on the rocks precludes them from settling, or, right now we don't know. And that's one of the questions that we need to try to figure out, because you can go to other areas at this depth and there is oxygen here. So I don't really know why they wouldn't be here.
Yeah,
But you
I
definitely
think
find
it's,
these sponge gardens that are pretty much all sponge. Go ahead, Lisa.
no, I was going to say, I think Pody's model predicted that there would be deep sea corals here,
Hmm.
but we're seeing mostly sponges, at least for now. What waypoint are we at, or which one are we traveling towards?
Yeah, we're traveling towards AC. We're kind of on the
Okay.
top of this
Yeah.
local ridge on the way there. Same strike. So it must be really hard to get information on larval recruitment and just recruitment in general for these species.
Yeah, so they put out plates and then they
Settling.
retrieve the plates to see what is recruited. But I mean, who knows if the plate is something that certain species even want to recruit on.
Right.
So they try different methods. One of the things that Liz Clark, she at Daisy Bank up at kind of mid-Oregon, they found this area where almost all the sponges were the same size. And so they think that trawling took took out all the adults,
Oh.
and now
left one.
this area, and now the area was recruited, over some length of time, but now they all look the same size. So here you can see like these yellow ones sort of look the same size,
Right.
but there's some smaller ones here and there, and then sometimes we hit a large one, but these are more like the white ones. They were more face-looking sponges that they were all about 15 to 20 centimeters across everywhere you could see. But there was no larger ones. Oh, but they grow larger, but there just was none around, or no smaller ones. So it could also be something like some recruitment events that bring them in.
Only
And
episodically.
then, yeah,
Yeah, when people talk about zonation in the benthos like this, I always imagine it must be mostly determined by the recruitment and those conditions. Or I mean, almost
That's true.
exclusively. So,
I mean, if you look at the rockfish, the rockfish, they have such great change in their recruitment every year. So one of the jobs I do is I actually scuba dive in the kelp bed and count baby rockfish.
Oh, cool.
And yeah, so one of the, like the low years, I usually do about 20 minutes a count per dive. And on a low year, I might count 4. And on a high year, I might count 10,000.
Oh my gosh.
So just
click,
the
click,
change in numbers. Well, the 10,000 is more of an estimate. The
Yeah,
4
I
is probably
hope so.
absolute.
Well, that's amazing.
Yeah, so they just have these years, and that's why rockfish are long-lived, and they have thousands to millions of larvae that they can produce, and over their lifetime. And so some of them hopefully will be able to survive. You get one of those good years and they survive, and then you have some progeny out there. But there's like the 90s was a really bad recruitment year for most years for most of the rockfish. The whole mid 90s were just no recruitment. Right when Lisa was diving with me.
Well,
Or
I
was it,
remember
no,
seeing
you were in the 2000s. Sorry, I gotta think of
yeah. I was gonna say I recall seeing canary rockfish with you and how cute they were.
it. I don't know if I go all the way to
How they-
cute, but okay.
No, they were very cute. How they blended in with the sand. I remember that vividly.
Yep.
So does it have anything to do with El Nino years up here?
It does. It has El Nino, La Nina. It has to do with predators out there, jellyfish populations, because that's what eats the juveniles, a lot of them.
The juveniles, not the larvae?
The ones that I see, so the juveniles are up to about, they start becoming juveniles at about 20 millimeters, so 2 centimeters.
Right.
So, it all depends on the size of the jellyfish, but they definitely eat juvenile rockfish. Some of them stay to be almost 10 centimeters before they come out of the plankton, so they get really large. Those ones may be able to withstand a jellyfish, but all of those, because we get 20-foot long jellyfish out here,
Right.
so eating a little rockfish isn't going to be hard for them. And we get the big brown ones out here. Their tentacles can go for a long way. Then the pelagias are another group. Oh, there's a cap shark. And yeah, it's not that the bell's that big, but their tentacles just stretch for a long distance. A 20-foot bell, I would be scared of that jellyfish. So actually the most potent ones, the Cuba medusas, the ones that you find in Australia are small, so they just pack more punch.
Yeah, small but deadly.
Yep.
Well, some of the ones like the Neomniopsis ctenophores and stuff that seem so benign to me, but those can do a number on larval fish is what I hear.
Oh yeah. Seems like everything wants to eat everything else out here.
Nature.
Everything's food. So I want to correct that the model doesn't predict seeing the corals until we get between waypoints AD2 and AD5, so much later in the dive, because we're now just heading towards AC. So maybe we'll just have to wait till we get to the Ds in the waypoints before we start seeing corals.
Roger that. I mean, I see some under the rock to the right. There's that fuzzy Clavularia coral, but there's just not many around. Oh, there's a mushroom coral. Right there, just went off the screen.
Big sponges
They
in the
got spoiled.
distance.
Yep. We got spoiled yesterday with the big Christmas trees. Is that one growing out of the other one, or is it just behind it? Oh, it's behind it.
See, that's why I think people who do ID work based off imaging are super skilled. You have to deal with like one orientation. Get used to the gestalt of all these things.
Yes, with just one image, it is very tough.
Yeah.
That's why the video helps a lot.
Oh,
Riley,
you can
gonna do a
see
quick
the
zoom?
dead one on the left there, falling down.
There's a lot of them. I see a
Yeah.
bunch of dead ones. And even the base of the tall guy is starting to die.
Oh yeah. Kind of looks like a cornucopia at this.
Yeah, it does.
It does. Maybe
A
there's
sort of
something in it.
a cornucopia for Halloween because it's dead.
Right, Riley, go ahead and come wide, please.
Well, that's big fin eel pout.
Yeah. Jeff saw it.
Yeah, just got a glimpse.
Got
So it
one
looks
line
like we're.
here.
Oh yeah.
Oh. Want me to hold the ship?
Let me see where it goes. I think it's flat,
Flat.
benign.
It seems like it's covered in sediment,
Sediment,
too.
yeah. Just can't be too careful.
Nope.
Yeah. At least anchored around the rocks.
Roger.
gonna stop the ship real quick?
Yep.
So how far have we traveled since the last transect?
We've come another 170, 180 meters.
So we could try to do a transect around here if we can find good terrain. We're not going downhill.
Yeah, this should be pretty flat for, yeah, at least 250 meters. Might start with a little bit of slope, but I think we're over this local max.
Yeah, it looked like that.
But
All right, Marley, you want to come actually a little bit wide? I'll get squared up for a transect. You good? Yeah. Go ahead and push in a bit. That's great.
and Jess, I'm gonna do 305.
305, Roger.
And if you can get as close to the bottom so that we can look for the moldy Swiss cheese as we go.
Roger that. All right, Ben, I'm ready for you.
All right.
These are the feather sea pens,
Great, thanks.
or the boa sea pens.
The feather boa.
They are.
Okay.
All right, starting transect.
Thanks.
Roger that.
Go.
Transect 5.
3, 2, 1, go.
Whoever gets there first.
I think we'll all arrive at the same time.
Yeah,
We better.
we all win. We all get it. We all get a cup or a medal.
Are there doubloons in it?
If we find that blue cheese, yes.
We've really
So
developed
people
our
who
own
are listening
proprietary
in,
language by now.
yes. But so for people that are listening in at this hour, we mean a sponge that looks like blue cheese.
It's a white and blue sponge, and it's small
Okay.
and round, and that's why it's called the moldy Swiss cheese, because you think of the Swiss cheese with holes, the blue spots would be the moldy part in the hole. But it could just be blue cheese. a lot of these sea pens here.
Sorry again?
There's just a lot of sea pens here.
Oh, that's yep. It's funny how obviously some species are undesirable. It's like, more sea pen.
Well,
Say
it's
that
better
again,
than
Ben?
just looking at.
No,
It's better than looking at the straight mud that we did yesterday when there was no sea pens.
yeah, I think it's cool.
And we're getting some more of these urchins.
Learning to share over here.
Yes, if you meant did you mean that sea pens seem to be a little bit more pedestrian compared
Well,
to
when they're
the
this
wow
numerous and
factor?
we're looking for the Swiss cheese, you know,
Yes, exactly.
it's all contextual.
Yes.
I would never say a sea pen is boring.
I was trying to understand what you meant.
Yeah.
They're like little question marks.
Yeah, they look like something from a Dr. Seuss book.
Yeah, I think so many things down at the bottom of the ocean or bottom of the ocean are very Dr. Seussian.
Susie.
Yeah.
Success.
Seussian landscape. There's a part of me that thinks about the tails of squirrels. They all have their head buried in the sand and their tails are sticking out.
Yes, like that squirrel from the Ice Age
Yeah, totally.
films. Yes.
Burying their
Trying to
acorns.
find the acorn.
That's the most vivid description yet.
Yeah, that's a good
that
one.
sticks with me.
Yeah, I'm gonna think about that.
Are they
from now on.
feather boa sea pens or are they squirrel tail?
So you guys got to find a new species so you guys can name it.
Squirrelinia.
Yes, and so are those white stalks? Are those dead squirrel tail sea pens?
No, those are actually other sea pens that are growing up.
Okay.
You can
That's
see the
showy.
polyps as we go by. Yeah, they're definitely not as big of polyps as you have on the feather boa, squirrel tail. Anthoptilum is the name, so we can just go that.
Okay, thank you. So I guess after transect, maybe we could zoom in on one of them if we see one after transect, so we can look at the polyps.
Sure thing.
Yes, that sounds good. And there's even a knocked over one of those to the
Yeah.
left.
So out of place.
Is that an eel pout coming up? Or no? No,
I
it's
see a
too
hake coming up.
Yeah.
Maybe a Pacific hake.
That is the one I meant.
Yeah. We have come 50 meters. Oh. So can those sea pens retract their polyps, or?
They can.
Yeah.
And they can even retract the stick.
Oh, really?
So
Into
yeah,
what?
so if you look at some of them, you can actually see the bulbous
Base.
kind of base that's under there, and they inflate that with water to hold them in place.
Oh.
So they can just release all the water, and then it goes down to a small nubbin.
Bridge.
So that hold fast, that's called a peduncle or a,
The peduncle.
yeah.
Jess is nodding. She's an expert on adhesion mechanism.
By no means expert. Very
Ooh, what
fun
are
word
Expert
those?
though.
here, for sure.
Dunkel.
Yeah, most of the sea pens are stick-like, but then and straight. But then you get to these feather boas, and for some reason they grow in a curlicue or a squirrel tail, whatever you want to call it.
Yeah.
It's gonna stick with me too now.
Yeah.
So here comes
Yeah,
your
I
wheel
think
path.
That's how it starts.
yes, I think we voted on squirrel tail. So
I'm glad it's sticking so well.
And the lone sponge in the middle.
Yeah. A rockfish under it too.
yeah.
Yes, it did look like a rockfish and not a thorny head. The thorny heads are flatter and spiny looking, and then we have an octopus in the middle.
Oh yeah, small little guy.
Trying not to get eaten by a squirrel.
They're natural predators, I assume.
Wasn't that Sandy on SpongeBob? Wasn't she a squirrel?
Oh yeah, but she had gear to be down here.
Yeah.
Well, they might.
She had
They might have their
right.
head in a bubble.
Right.
They're all sleeping and have their head under there. That's what it is.
This is a very good hypothesis.
we need
Yeah,
to
you can
pull
write
one
the
up
paper,
to
though.
check it out.
What is that?
It's interesting how
Yellowing mound.
it's switching over now, though. From all feather boa to now
The
more
pen.
sticks.
Yeah, it is
Yeah.
interesting. It's way more gradual donation.
And what's the name of the straight sea pen?
Straight sea pen.
Okay.
I could not
The
tell you right now.
non-feather. Okay.
It's
I'll just call it the
sea
straight.
pen.
Okay.
The A squirrel.
Yeah.
I was gonna say bald squirrel tail. Hairless squirrel tail. Rat tail.
The frightened
We could do the
squirrel
Halloween one. The skeleton squirrel tail.
tail. Yeah, exactly.
But Meredith she's one of the ones who will attempt to call out what they are. Like with many other similar looking corals, I usually will just call them sea pens unless I can get an absolute ID on them. She's more willing to tender a guess on it. And I see sponges coming up. So that was just a whole field of corals. And now we move back into the sponges.
Looks like we got another line here. I'm gonna change my heading here. Like it might be benign again. Yeah. All right. I'll go back to my original heading.
And there's some corals right there. We had to cross the line to get to the corals.
Yeah.
Yeah.
The Swiss cheese will have 4 lines.
Scales.
All right, Ben, go.
Yep.
I'm gonna turn my heading here.
Thank you.
And more of this clavularia coral, this fuzzy stuff on this dead sponge right here
Mhm.
on the bottom. And how far have we traveled so far?
Yeah, so we've come about 120 meters, so 80 meters to go.
Roger that, thank you.
A small consolidated patch in a field of squirrels.
Yes.
The squirrels have surrounded those sponges and they're closing in.
It's
An
really
army of
harrowing.
squirrels. Yeah.
Yeah, we're like right in the middle of these two local features. Kind of like a little valley.
That makes sense.
I was just about to start passing them out. I was going to wait till it was 6, but we're right there. Coming
Are
your
these
way.
the chocolate covered
Hey, Kim.
coffee beans?
Kim. This way.
And how do we get those?
Oh, did you guys
I
hear
know.
our candy talk?
I was just going to
Yes,
ask
we
if
can.
Tom and Lisa, I was going to ask if you guys are coffee drinkers. What's your stimulant for the morning? 4 a.m. watch?
Thanks, Riley.
I just wake up. I actually don't like coffee, so.
Oh man, au natural.
No espresso beans for
Yeah.
you.
Actually, I don't mind having those, but I don't know, drinking the drink, that's never appealed to me. Never tasted good.
I'm not completely au naturel, but I drink an Earl Grey tea. I'm not a coffee drinker either. It sounds like you guys are hardcore over there. Do you take it in your veins or what?
I do instant coffee, which is just as bad.
Oh, there's a knocked over feather boa, completely out of the sediment. Or the tail got cut off the squirrel, one or the other.
So it was
Mound.
6 o'clock on the dot that you guys held out. That's pretty good. That shows some restraint.
Gotta pace ourselves.
Yes. So who is the chocolate covered coffee bean person? I was going to ask, but you guys gave yourselves away.
Yeah, so that's me. I'm a huge coffee drinker. This is Marley. And when I came on the ship last year, it was my first time sailing on Nautilus, and I wasn't aware that the coffee they provide on the ship is not really the very best coffee ever.
So just call out the brand. Is it Folgers or what?
I don't know.
That would be an insult to Folgers.
It's I don't want to speak negatively of any aspect of this ship, but the coffee was not up to my personal standards.
Okay, I get it.
So I only survived and stayed awake because our ROV pilot, Greg Diffendale, brought an enormous amount of chocolate-covered espresso beans. So I returned the favor this year and brought a big supply, and also brought my own bags of coffee, lots of things, supplies.
Whatever it takes, right? I get it.
Yeah, totally.
Yeah, it's a difference being on the ship than being at home. I can go get anything I want.
Go play with your cat.
Or I can kick it away too. That's always fun.
More likely. My cat always sits in front of the screen I'm using.
She comes to my chair and just claws at me in the chair to get petted, and I'm like, no, go away. Oh, there's one of the black coral crabs on a sea pan, since there's no black coral around here. And you can see some of these tall stick sea pans actually have brittle stars wrapped around them. Of course, none of them are there while I'm seeing. And there's one of the squirrel tails pulled completely out.
5-0.
Oh yeah.
Those brittle stars seem to do well in a lot of different host animals.
Mm-hmm.
Are they pretty, like, our species pretty specific about what they're on, or I don't know, the selection?
They definitely go by depth, and I do think that some of them would like more fine mud or more thick mud, but that also could be a something to do with depth, because you get finer mud when you're deeper. Because if you get to about 80 meters, you have these ones that grow like this, and then they have basically big long whips that grow off the top. That can be a meter or two long.
Oh, wow.
But I've only seen those on the around the 100 meters, 80 meter ones.
So are these transects directly to facilitate like counting and quantitative aspects of looking at the video?
Exactly.
Yeah,
So
Okay.
someone is going to go through and count all these,
that's
the
what I
two
was thinking.
species. Yep. Someone's going to go through and count all these. And it's going to be different than the mud transects from yesterday,
Yeah.
where there wasn't many C-pens and they will go fast. And then this one will be much slower in counting. And then we take all that data and since we know how far we've gone, we use the lasers to determine the width of the transect.
Oh,
We can get an area, and then from that we can get densities, and we can compare those densities from not just this area, but along the coast. Anybody who's done densities before, we can compare them to those kind of areas. We also look at the habitat. So here it's going to be just mud for this section. And so you look at what's growing where and try to determine where there's hot spots, why something might be there. We have a lot of the CTD data that we can use and some other environmental data to be able to put into models like Matt Pody does. But then we can also just look at fine scale areas like sitting in there, a 10 by 10 area and come back year after year and look at that and see how it changes over time to look at assemblage changes, things like that.
really cool. It's too bad software can't do more for you to automate some of the counting and stuff.
There is some out there. And so like these C-pens, we probably could get some of that, especially the feather boas. That's a pretty easy algorithm that might be able to be figured out. The stick ones, because they're definitely on this mud background. They're pretty easy or this mud bottom. They'd be easy to pick out of the background, and probably the white sticks too.
So
but
actually,
then we also look like other data.
Sorry, yeah, we
We're
just
at the
have
end.
come 200. Yeah.
All right. Well, we can stop then.
All right. You want to hold the shift?
Yep.
And then I know Lisa asked before if we could zoom in on some of the sticks.
Yeah, I can sit down and then do a change over here.
Thanks for remembering.
Get a nice tight shot on these guys.
If we come across more of those sea urchins, could we also get a couple samples of those for microplastics?
Yeah. Yep.
Really
I
good
see one in the upper.
Oh yeah, now you can see it. That is amazing. So the polyps just sort of disappear as you're zooming by, and then compared to the very feathery, bushy tail of the squirrel.
Yeah, if you look at a polyp from the feather boa, and then you
Yeah.
look at one of these polyps, they're completely different
Yeah.
in size-wise, at least.
But the
There's a little
idea is the
baby.
same.
And this may have eggs in it, all the little white dots
Oh, really? Right at the base of the polyps?
right at the base of the polyp, yep. Maybe not. It's hard to see.
Is that how they would be fertilized or released? Right on. Right.
Yeah, that's how they would, yeah, they're, and then they work them up and release them.
Oh, I see.
Thank you, that's good.
Thank you for doing that. That was very interesting.
That's what we're here for.
Yeah,
And
you can
now
see
that we've
them.
seen them up close and we're further out, I can now see the polyps better. But previously, they just sort of looked like nude white sticks.
And while we're here, if we could sample one of these sea pens, the feather boas, because it's on our list.
Nice, I didn't realize it was on the list.
I just figured I'd look it up while we're looking at it.
Maybe we get the whole thing, right?
Yes. And since I see the requester is Taylor Ann, we might as well try to get it.
Sorry, what was the request for the urchin that we just passed?
And one of the feather boas or squirrel tails.
Okay.
So if you go forward, it looks like there's an urchin and a feather boa together. To the right now.
Do we want to fire a Niskin bottle too then while we're at it?
Have they fired one yet?
We
I
have
see that
fired one.
Yeah.
So I take it back. They actually don't want this sea pen. They want a different species. It's still an Aethoptilum, so we don't have to collect that, but we can collect the urchins.
Roger.
Awesome.
And I'm looking, we've only collected one Niskin bottle.
Is it possible to collect this through a slurp, or would it be better to do a grab?
Is there a preference?
I think Taylor Ann prefers a slurp, but if that's not possible with this urchin, BioBox is fine.
We can have a look as to how big it is with comparison to the hose. We just don't want to get stuck in the hose.
All right.
But we can put the hose next to it and see how it goes.
I don't think it's going to run away very fast. And while you collect, I'm going to walk away for about a minute or two to stretch my legs, and I'll be right back.
Roger.
Roger. have your joy gain turned down as well. Or sorry, grip force.
It's an
Yeah.
intimidating gesture.
Big jaws are no.
So did you guys put the coral clippers on it now?
Yeah, we put the coral cutters back on. Doesn't help when it's right below the brow bar, does it? I'm gonna rack you full back there, Kim. That's all right. I'm gonna rack you back. That's good. These jaws, they're not to be trusted. They're just so big. I can calibrate it after we're done with the sample if you'd like. Roger. If you notice it creep, we can take it out of the continuous wrist and then it won't creep so much anymore on you.